Good afternoon, everyone, and thank you very much for joining us for this HDSA research webinar. I am Leora Fox, Manager of Research and Mission Programs at the Huntington's Disease Society of America. And in just a few minutes, I'm going to introduce our speaker, Dr. Peg Nopoulos. But first, I just want to remind everybody about how we run these. So questions will be answered after the presentation. You can send a question anytime during the presentation, however. You'll see either there's a little bar at the bottom or perhaps at the top, there's a drop down in Zoom. You can click on the chat function, type in your question, hit send, and only HDSA is going to be able to see your question, then we will ask those questions to Dr. Nopoulos at the end of the webinar. We are also going to be recording this webinar, so to view this we usually try to put them up within a few days to a week, and you can go to hdsa.org/researchwebinar, or you can go to HDSA's YouTube channel. So at the top of our homepage, there's this little YouTube button. You can also search us on YouTube, and that way you can view this or send it to someone who might not be able to make it today. So without further ado, I want to introduce our speaker, Dr. Peg Nopoulos. She is a professor of psychiatry, pediatrics, and neurology, and she is the Paul W. Penningroth Chair and DEO and Chair of the Department of Psychiatry at the University of Iowa Hospitals in Iowa City. Dr. Nopoulos is a physician scientist. She takes care of patients with Huntington's disease in Iowa's HDSA Center of Excellence. She's worked there for the past 15 years, and her research focuses on the study of brain and behavior using imaging techniques like MRI, and she's particularly interested in studying the effects of the HD gene on brain development, and she's the director of the Kids JHD program and now the Change HD program, which she's going to talk about today, and we're very excited to have her. So thank you for being here, Peg. Now we'll hand it over to you.
Okay, you are very welcome. So, can you see that yet?
Yes.
Okay. I'm going to do the, let me do the display settings again and duplicate slideshow. Okay.
There you go.
Good. All right. That's just a beautiful shot of the College of Medicine buildings here at the University of Iowa. But I'm going to start with an outline just to kind of pace us along about what we're going to talk about, because they're kind of new concepts. But I'm going to talk a lot about human brain development because you have to have a basis for that before we talk about the gene. Then I'll discuss a little bit about the gene and how the gene affects brain development. And then we'll talk a little bit about how this is important, and I think what everyone's mind is on these days is in Huntington's disease, you know, the concept of gene therapy and how does this, you know, change this or in any way. And then I'll talk about our new program. So you'll hear me talk about Kids HD, which is our former program, and Change HD is what we have going forward. So the first thing to talk about is the human brain, and just to give you some concepts, I'm going to be showing some images and just want to get a sense 3D of what we're looking at in terms of a brain. And then when we slice it open, or when we take a picture of it, like with an MRI scan, it's in what we call this coronal view. So I'm going to be showing some slides of MRI scans, and that's kind of the orientation. If you think about looking at, you know, this face here, that's what the brain is going to look like when we slice it in that area. And so here's an MRI picture of that. So that's the coronal view. So you're kind of looking, if you were looking at someone's face, this would be the picture of their brain. But the basic part of the brain is this on the left, which is a brain cell, also called a neuron. And the parts of the neuron are important because they make up different tissues in the brain. So this area up here with the green dot, this is the cell body. And these are called dendrites. What these are, these little things that come out and spread out. And then you have a very long, what's called an axon. and it's covered with what's called myelin, which is a very fatty tissue. But that is the axon and then the myelin sheath. And at the end is the axon terminals. But most importantly, when we look at this scan over here, we can see that there's essentially gray and white in this image. Pretty much everything that's gray matter is these dendrites. It's the cell bodies and dendrites. And then the white matter is, this white matter in here is the axons. It's the it's the axon sheaths. So the tissue within this scan, the first is the cerebrospinal fluid. So that's the very dark areas that you see right here and right on top of the brain. This is actually the fluid that surrounds the brain, and it's the fluid that surrounds the spinal cord. And so when you have a spinal tap, that's the fluid. So for instance, in the gene therapy trials, getting the drug into the spinal cord goes into the CSF, and then it bathes the brain. It goes into the brain tissues. And then in addition to the CSF, there's that gray matter. So it appears on an MRI scan as darker gray. And you can see that it's all over, but it's on the top, but there's also big hunks of gray matter in the middle of the brain. And then the white matter is, once again, all of this tissue that looks bright white. So those are the tissue changes, or the tissues that make up the brain matter. When we put all the tissues together, in general, we call them intracranial volumes. So this is the cranium. So when we say ICV, we're talking about essentially just total brain volume. That's a term that you'll hear me say, ICV. The other thing that's important is, I'd mentioned this, in that gray matter comes in both what we call the cortex. So it's all this squiggly on the outside. Cortex actually means bark, like the bark of a tree. It's the outside of it. But there's also gray matter inside. And the one area that you may have already heard about, because the striatum, which is made up of the caudate and the putamen, this is a region of the brain in which, for Huntington's disease, is where the primary issue is with the etiology of the disease, with the that's where the origin of the disease is in the brain, is in the striatum. So that's a really important area to think about, and I'll be discussing how that grows and develops. So one thing I want to bring up is this notion that there's an analogy between a neuron and a tree. And you can think about those dendrites as having branches. So that portion where you have all of these little spindles coming off, you can think about them as the branches of a tree. And then the axon as being the trunk, and the axon terminals being the roots. And that'll be important when I talk about the concept of pruning, and when you prune your branches. But I want you to think about a neuron like a tree. So here's some beginnings of how we look at the brain in the context of brain development. This is actually a slightly different section of the brain. This is actually looking on top rather than looking towards the face. But this is MRI scans at birth, at 1 year, and at 2 years. And over here, we're just looking at the volume. But I think you can see very easily that between birth and 1 year, there's a dramatic change. The brain increases by 100%. So it's a doubling of the size of the brain. So that time between birth and 1 year is a very dramatic change. Now, between 1 and 2 years, you can see there's a little bit, there's still growth, but it's not a lot. It's this time between birth and 12 months that there is some of the most dramatic changes in just brain volume. Then onto this next graph, this is childhood brain development. So we've been studying children for a long time in our lab. And here's that term I used, intracranial volume, which essentially just means total brain volume. And now we're looking at a large sample of people who are between the ages of about 6 and 25. So this is what we call childhood, so to speak, and I'll explain why childhood goes all the way up this area. But when we look at the average growth between 5 and 25, that's this red line here, you can see that there's not a lot of total brain volume growth. And as a matter of fact, total brain volume by 6 years of age is pretty much adult size. 95% of the volume at 6 years is you only have 5% to go at this time. So the brain volume doesn't change much. However, what really changes dramatically during that time is the tissue inside the brain. So this is an example of that same age range between 5 and 25. And on the left is cerebral cortex volume. So that's the gray matter that I said was on the bark or the surface of the brain. And this is the cerebral white matter volume. But you can see here between ages 5 and 25, this is normal, healthy children. And you can see this red line is over time, there is a substantial drop in the volume of the cerebral cortex. This is normal. This is absolutely normal. And it happens most steeply over the period of puberty. At the same time, even though the cerebral cortex is decreasing in size, the cerebral white matter is increasing in size. So even though I showed you that the total volume doesn't change much, what's happening is that the tissues within the brain over this age range are changing pretty dramatically. What we call pruning, remember I'd mentioned to you about the tree, what happens is that the gray matter gets pruned. And this is called program synaptic elimination. Essentially what this means is that the dendritic branches, just like you would prune the branches of a bush or a tree, they get cut back. And then with the white matter volume, what you get is you get an increase of that myelin sheath, that white matter. So once again, these are pretty dramatic changes that occur during childhood years. So the tissue remodeling, it starts oftentimes with the pruning at the beginning of adolescence. And once again, it's the pruning of connections. We're not losing neurons, okay? So during adolescence, you might think that there's neuron loss, there's tissue loss, but it's not neurons, it's just the connections. So this is a picture of, the neuron with all these dendrites, and then you can see here, there's just fewer dendrites. And this is important because it's normal. So what happens is during adolescence, you prune, just like in a tree or a bush, you prune the connections that you don't need, and then you strengthen the ones that you do. So that's what happens in the brain, is that these are still maturational, developmental changes. And all of you who are familiar with the phenomenon of adolescents and teenagers, there's a lot of change going on in the brain that is the kind of the biological explanation for a lot of the behavior that happens in adolescence. But these are normal developmental changes. So pruning, which is good for the brain, it results in a decrease in volume. So once again, these developmental changes, I say between 6 and 25, they're actually lasting a little bit longer than that. But the changes are mostly tissue distribution, right? So I told you that total brain volume is pretty much adult size by age 6. But between 6 and 25 or 30, you're having all of these tissue changes inside. They could be termed late maturational changes. Sometimes I refer to those changes as development because your brain is still developing. But you could also think of them as maturational, right? So that they're changes that are happening during adolescence. But the most important thing to remember is that the human brain takes a long time to develop. At least until 25 or 30, these maturational changes, the pruning and the myelin, this is a prolonged period. So it's really important to think about the fact that brain development is a normal process that takes decades. So now on to the next topic. Most of you probably already understand this, but it's really important just to make sure that everyone is on the same page about the basics of the gene and what we call it, or HTT. So the genetic code is DNA. It's made up of nucleotides, and we have 4 letters in our DNA code, and 3 of those letters make up a word, which makes up an amino acid. And so we have these combinations of these 3-letter words, AAG or CTG, and then each one of those amino acids gets strung together to make a protein. So you have the gene up here. Each one of those 3 letters make up an amino acid, and amino acids are then strung together. And that protein is essentially what does all the work in the cell. What happens in Huntington's, so the gene is called Huntingtin, and we call it HTT. That's just the kind of the nickname for it. So when I say HTT, what I'm just talking about is the Huntington gene. So what happens in this gene is that there is a DNA stutter, meaning that we have this amino acid, glutamine, which is this code, CAG, repeated multiple times. So the gene normally has that. So just to remind everybody, every human has Huntington, okay? It's a normal gene. And every human has repeats. So it's normal to have repeats in this gene. Most people in the normal population have repeats around 9 to 25. But what happens with Huntington's disease is that if there is too many of these repeats, that's when disease develops. So a couple of terms I just want to mention because I'll be using them later. We have another way of naming them. So repeats below 40, some people would say below 36, but we call this gene non-expanded, GNE. And that means people with repeats less than 40 are not going to develop Huntington's. Repeats above 40, so that's going to be called gene expanded. So that means that there is a larger number of CAG repeats. It's 40 and above, and that's determined as GE. So I think all of you are probably familiar with the fact that there's a spectrum as well, so that the longer the CAG repeats, the earlier the HD onset. So everything with this gene, it's really important, every CAG repeat counts. I'd mentioned that 36 to 39 repeats is sometimes called intermediate, but repeats in that range often end up in what we call late onset or Huntington's disease that occurs between 70 and 80 years old. And you can see as you increase the number of repeats here, so going to the left in this middle bar, 40 to 49, over 50, over 60, you get the onset earlier and earlier, such that in general, if you have repeats greater than 60, you often get what's called juvenile onset, so onset before the age of 20. But just a reminder that everything is a spectrum and that each CAG repeat means something in the function of this gene and this protein. Okay, on to the next topic. So effect of HTT on brain development. So this is where things get interesting. So we know that this gene affects brain development. How do we know that? One reason is if you've ever heard of the term knockout. So one way for us to understand how a gene works is that we take a mouse model and then we knock the gene out and see what happens. Because if we knock the gene out and then we study that animal, we can see, wow, well, without that gene, this happens. And when we do that, we see that the brain doesn't grow right. So we know that the normal gene is involved in brain development. There's been lots of other scientific experiments to show that the normal Huntington gene plays an important role in the health of brain cells. So the question that we had in our lab was, if the normal gene affects brain development, what happens in people who carry the mutated gene? Or what happens in people who carry a gene with a CAG expansion, those gene expanded people? They have that gene from birth on, And if the normal gene affects brain development, how does the abnormal gene affect it? So that's the question that we had going forward. And this is just a schematic of a figure of how we might think about Huntington's disease and how we think about degeneration. So what I mean by the term of neurodegeneration is that we all know in the context of this disease that the disease manifests by brain cells getting sick and then dying. And degeneration essentially just means progressive over time. And at the top is what we call the classical concept. So on the left, we have this normal neuron. There's the dendrites, there's the axon and the myelin sheath. And then after it grows up normally, you have a disease process. So the MHTT, that just means mutant Huntington. So that's an expanded Huntington. And the concept is this expanded gene product is toxic to the brain cell, and so it degenerates and dies. That's kind of the classic concept of neurodegeneration with this mutated gene. But in our lab, we think about this concept of called developmental concept, meaning that the disease process actually starts with abnormal development. So if you can see this cell here, he's not quite as well developed as this neuron up here. He doesn't have as many dendrites. He's a little bit shorter in his axon. He doesn't have as many axon terminals here. So you start out with a neuronal cell that is not very well developed. So it may have been affected by the mutant Huntington. But everyone knows that this disease doesn't manifest until later in life. So how is it possible that when you have an abnormal neuron, an abnormal area of your brain, how do you not have the disease? Well, what happens is that you have some compensation, some other area of the brain takes over. And that's what's called, I know it's a really weird statement, but it's called a mutant steady state. It means that even though this cell and this group of cells is not formed well, it can still function because there's some compensation. But what happens eventually is that this neuron gets tired. And so it's being compensated for during childhood, but eventually, whether it's just the maturational process or aging, or maybe it's this toxic protein, but eventually that little buddy gets tired and it degenerates. So it's really a different concept. It's thinking about the beginning of the disease happening with brain development rather than this process happening later. So we were interested in looking in how Huntington affected brain development. So this was our original study. It's called the Kids HD study, and it's a study of children at risk. So we started with subjects that were 6 to 18 years of age. We had a few people that were above 18, but they were all at risk. They had a parent or a grandparent with Huntington's. We flew them in from all over the country to the cultural center of the universe. It's my home, it's Iowa. So everyone came to Iowa. It's a one-site study. And then we also had healthy controls. So these were children with no family history of HD. And we looked at brain function. We looked at thinking tasks and motor tasks, and we got an MRI scan. And then all subjects donated DNA. This was for research purposes only. This is very important because these genetic results were not provided to anybody or to us. So they were for research purposes only. And anybody below 39 was GNE, and repeats above 40 were designated GE. So once again, those are the terms that I'm going to be using, GNE and GE. And this is just a table showing how many we had in each group. There was quite a bit. We had 138 controls. We had 117 gene non-expanded. We had 81 gene expanded. A lot of these children came back for repeat visits, so we had multiple visits. And you can see here the age range, 6 to 25, but on average, most of these groups were on average about 12. But it was a large range in terms of over time. And so this is a graph that I'm going to keep up for a bit because it's really kind of the most important graph to think about, because this is the way that we looked at the development of the striatum. So remember, the striatum is the piece of gray matter in the middle of the brain that makes up the caudate and putamen. And we know that this is the region of the brain that is most important for Huntington's disease. So what you're going to see here on the left-hand side is an adjusted volume. So this is just how big the striatum is. And on the bottom here, you see the age. And I want you to first look at just the green line. So the green line is gene non-expanded. So these are kids who are not going to develop Huntington's disease. And you can see early on, their striatum grows until about adolescence, and then they start the pruning process. Remember that discussion about how in adolescence, you start to see a decrease in the volume of gray matter? So the GNE kids grow their striatum normally, and then they start the normal process of pruning. So that's the shape of the green curve. Now look at the red curve. This is the kids that are gene expanded. And what you'll see is that already from the very beginning, these children have abnormally large striatum. So as young as we could look at, they start out at a different point, which likely means that they got there several years before this. The earliest is age 6. So early on, their striatum is larger than normal. And then you could see the pattern, what we call trajectory. This is just how their striatum changes over time, how it develops. It's a very different pattern. From the very beginning, it starts out too large, and then it continues to decrease, so that you can see here, you know, sometime pre-adolescence. Now, at this point, if you were to measure the striatum, it would be smaller than normal. So you can see how it just changes differently over time. So this is really important, and this is really kind of the culmination of about a decade of collecting this study. And this in general was everyone together. It looked at just the average change that we see for these two groups. But then we wanted to see whether or not the repeats mattered. And they do. And what I mean by that is, if you can see here, this GE line, this is what I showed on the previous, if you can just, I'm going to go back to this slide. So this is the gene expanded, that's their curve. But we took that and then we broke it up into CAG repeats. And in particular, above 50, we could see that the greater the CAG repeats, the more different the trajectory was. So if you started with CAG repeat of 50, this was the line. But you can see here with each CAG repeat, as you increase it, that trajectory becomes increasingly abnormal. So if you think about the highest repeat here that we have of 59, remember that all of the gene expanded kids start out with an abnormally large striatum. But if you're at the higher CAG repeats, your striatum is at the highest, and then it falls the fastest. So not only does the gene expansion affect the striatum, but it's also dependent on how many repeats you have. The more repeats you have, the more dramatic the effect is on the striatum. So once again, I'm going to come back to this notion that the disease process starts as abnormal development. But if the striatum is abnormal, why are these kids not having any symptoms? Because these are kids and young adults who are not going to get sick for decades, okay? These are not children who are currently sick. These are children who are going to develop it later in life. And remember what I said about the fact that they don't have any symptoms based on compensation. And what I mean by that is that there's other parts of the brain that probably come to the rescue of the striatum. And we think that part of the brain is the cerebellum. So you can see here in red on the left, this is the area of the brain that is the striatum. But here on the right, this area here in the back of the brain is a relatively large area, and it's called the cerebellum. This area of the brain grows together normally with the striatum. So normally there's a balanced circuit. If this grows normal, this grows normal. But what happens in Huntington's is that when the abnormal circuit, so when the striatum doesn't grow right, then the cerebellum can compensate. So initially, when the striatum is not developed well, you have an abnormally imbalanced circuit. But if your cerebellum can compensate, then you're able to have a balanced circuit. And this is what happens, this far right. This is what happens lifelong in children who have a gene expanded. So their striatum isn't developed right, but their cerebellum kicks in, and they don't have any symptoms until later in life. So we think that even though the striatum doesn't grow right, the cerebellum helps out. And so this is just a funny cat picture that suggests that a lot of people are surprised by the fact that the cerebellum is involved, because the cerebellum is really considered to be an area of the brain that is spared from Huntington's disease. But we think it's spared because it's doing a job of compensating for the striatum. And I'm just going to show one picture here, and I'm not going to go into details of how we do this, but these are just MRI pictures, and it's a comparison of the connectivity, so how strong the connection is between the striatum and the cerebellum. And if we see blue, we compare the gene expanded to the gene non-expanded, and these areas of blue indicate that these children have stronger connections between their cerebellum and their striatum. So it's just supporting evidence to our theory that the cerebellum is overconnected, and that it's compensating for in these children who are gene expanded. So how might this affect gene therapy or other treatment? So I'm going to show you not brain morphology, but this is a test that we call verbal fluency. And as any of you who've participated in cognitive studies, you probably know this test well. It's very simple. Name all the words you can think of starting with the letter C, and you get 60 seconds. And excuse me, you can see that over time, the verbal fluency score on the left-hand side here, it increases. So this is something that normally develops over time. It actually peaks near age 30, a little bit older than age 30. I'm showing some of my lab members here in the lower right-hand side. So Ellen Vanderplas and Jordan Schultz, who've been working very hard on this. But you can see that this verbal fluency score, and this is for all the people here. I just wanted you to show that this is something that takes a long time to develop. So as I mentioned before, it takes a long time for the brain to develop. This is just a brain function, and you can see it takes a long time. But when we break that same trajectory down by repeat values, we see that once again, repeats mean something, each repeat. So you can see somebody with a 38 or a 40, this is the same thing, verbal fluency scores, they are still growing that skill. They haven't even peaked in that skill because this only goes to age 20. But for people who have a longer repeat, 44, 46, 48, you can see that they are not reaching the maximum potential of their verbal fluency skill. And it suggests that they're already having difficulties with this score. And we would also know that the longer the repeat, the earlier the onset. So it's possible that at this point, that they're already being affected by the mutant Huntington. So when we think about gene therapy, I think we need to think about potentially when we would offer it. So currently in gene therapy, the studies are being done on subjects who already have disease, typically early in the course. And what we want to know from these early studies is, do they not degenerate? In other words, they're already sick, but do they not get worse? Eventually, we're going to want to prevent disease. So knockdown of genes for prevention is the next step, right? So if gene therapy works to slow the disease, when do we give it so they don't even get the disease at all? And I think it's important to consider the fact that we need to think about brain development, and we need to think about the fact that even the mutant form of the gene is affecting brain development. So for instance, if we were thinking about knocking down the gene at, say, age 16, remember that this gene is still driving brain development. And so if we were to give therapy here, we might prevent full brain maturation for this skill. On the other hand, for people with longer repeats, they're already showing effects at 16. So maybe for longer repeats, we might want to intervene early. So once again, these are all just suggestions because this is the first time that we've evaluated this data, but this is a possibility of how we might think about prevention trials, when to intervene, how to intervene, what age. And I think it may be very dependent on the repeat length of the mutation. So Change HD is the new branding of Kids HD. And what that is an acronym. So child to adult, neurodevelopment, and gene expanded Huntington's disease. So that's what's Change HD. So what we did is, this is funded by the National Institutes of Neurologic Disease and Stroke, NINDS. And so we had our Kids HD program for 10 years. And when we renewed it, we changed it to Change HD. So the NINDS approved our period project, which is, it just started recently. We haven't quite started running people through yet, but we're increasing the age range. So the eligible participants are young people at risk for HD, and we're increasing the age. Before we capped it at 18, now we're going to age 30. And the reason for that is because I remember the brain takes a long time. So we want to model all of brain development. And so just a couple of things, the new things for Change HD, it's essentially Kids HD. So we're doing very much the same thing we did previously, but we also have more computerized tests. We're going to do it multi-site. We're going to use some sensitive measures from our colleagues developed in Germany from Dr. Ralph Raumann. We also have collaborations with Dr. Wild and Lauren Byrne in UCL because we're looking at a biomarker called NFL. And once again, it's at-risk young people ages 6 to 30. So that's why we changed it from kids to change, because it wasn't just kids if we're going to go up to age 30. So very briefly, these are the sites. So it's University of Iowa, it's University of Penn and CHOP, which is Children's Hospital Philadelphia, Columbia, Houston, and UC Davis in Sacramento. And very briefly, just at each one of these sites, there are HD experts, there are imaging experts, and then there's boots on the ground people who are the research coordinators. So that's Philadelphia and Columbia, University of Texas, Houston, and Sacramento at the University of California, Davis, and then University of Iowa, where the primary spot is, where we have the biggest team. So I just wanted to thank everybody this is my lab, but to thank everybody who's ever participated. I mean, all of the families who've come to Iowa over the past 10 years, we're so grateful because all of this data comes from their hard work. And on the bottom are just all of the other organizations that have been helpful in supporting everything we do. And just one final slide to show you some of the specific information if you are interested in, if you are eligible or if your child is eligible, that you could call us. We also have an email address as well as a website address. So thank you. I'm hoping that leaves us time for some questions. Do I stop share now then?
Yes, there's plenty of time for questions. I am not seeing any come into the chat yet, so we will wait a little bit on that. But I can start with a couple questions as people start to write in. I thank you so much for this presentation. It was very clear. I was especially surprised to hear that that by 6 years old, 95% of the brain volume is already there, and also some of the findings about the cerebellum compensating.
Right. I think
But,
that,
okay, yeah, go ahead.
So I think it is, this concept of the brain taking 30 years to develop is not necessarily that well known. I mean, scientifically people know it, but I think in terms of our society, especially because, we define adulthood at age 18, that is not a biologic construct. That's a societal construct. So, if we really had to define when childhood ends, it wouldn't be until about age 30. But yeah, you're right. At 6 years of age, even though the volume is mostly adult size, there's still a lot that happens to those tissues during adolescence. So it's a very dynamic period.
Well, we are getting some questions in right now. So the first question is, are there male-female differences in verbal fluency development based on age and CAG repeat?
That's a great question, because there are a lot of sex differences in the brain, and sometimes they're hard to ignore. We have not looked at, so in general, females typically have better verbal skills, comparatively so. We have not looked at whether or not that there is a sex-specific change. In other words, is it, I would expect that the HTT, so the Huntington's effect, would be similar to males and females. And when we look at that, that's essentially what we see. So even if there's a normal sex effect, there doesn't seem to be a difference with the Huntington gene. It seems to affect the males and females equally.
Okay, thank you for that. Another question we have is about the enrollment period for the new CAG study. So maybe in general, you could talk a little bit more about how long is the study going on? Is it possible to participate if you live far from one of the sites that you mentioned?
Sure. So, previously we brought everyone to Iowa. So it didn't matter where you lived, we'd bring you to Iowa. And so same thing, it doesn't matter where you live, even if you're far from a site, we pay for travel, okay? So you can either pick a site that's closest to you or pick a site that you're interested in. We don't force what site you go to, but there is travel paid for you to get to a site. So whatever site you want to go to closest, you can get to one of those 5 sites. And in general, depending on who's coming, so for instance, if it's a minor, then we pay for the cost of the minor, and then of course a parent, because the minor can't travel separately. So very much like what we've done in the past 10 years, we brought everyone here to Iowa, same thing. We will make sure that people get the opportunity, but if they happen to live in California, they can go to, Sacramento instead of coming to Iowa City. Same thing if they live in Philadelphia, they can go there instead of Iowa City. But it's up to the family. And in terms of the time frame, so the grant started a few months ago, but it takes a lot of time for us to. So right now we're in the organization phase where we're making all the hires for all of the coordinators. We're training everybody on how to do the same exact protocol. Same thing with all the MRI scans. We have at Iowa, since we're familiar with this study, we will be asking people who have previously participated to come back. And we've already had a few of those, but the new protocol will be at the beginning of March. And so we'll have our first subject in the new protocol in March. So the other sites are going to be soon to follow, but we're really kind of just wrapping up some of the training sessions. And it'll be going for 5 years. And for the most part, we would expect people to participate more than once. They're not required, but in general, we like to have people come back on more than one visit. typically annually, but it, like I said, we do our best to work around people's schedule.
I'll add to that this study is listed on hdtrialfinder.org, and as the sites are really open to participants, we'll continue to update that.
Great, thank you.
We have some questions around age of genetic testing. So in general, children are not usually eligible to be tested until they're 18 unless they're showing positive signs of JHD. And so a couple of people are wondering whether the participants in this study are generally showing symptoms, or if this is usually kids who have lower repeats.
Yeah, so it's meant to study children who are not manifesting symptoms. And so in general, when we talk to parents who want to enroll their children, if they have not received the diagnosis of juvenile onset, they're eligible for the study. We have a separate protocol. If the child has been diagnosed with juvenile onset, they can participate, but and they would have to come to Iowa City, but we have a separate protocol. Change HD is meant to evaluate people, before they're ill. And so if in general, it's children who are not manifesting symptoms. But if they've already been diagnosed, they can participate in a slightly different protocol.
Great. We have another few questions that talk a little bit about variability of CAG repeats. Someone has mentioned that their husband has a 50 CAG repeat, but has just recently at age 43 been affected. Another similar question, which doesn't seem to match up with some of the, the diagrams showing sort of the length as a function of the age at which people are diagnosed. So maybe you could talk a little bit more about that kind of variability.
Yeah, sure. So there's certainly that even though there's a strong relationship between how long the repeat is and when the onset is, it's not a one-to-one relationship. So absolutely, there's going to be people who have a long repeat who don't get ill until later. Same thing with the other way is that, not everybody with JHD has a repeat over 60. Sometimes they could be shorter than that. So there is variability. And a couple of things that the scientific efforts into evaluating that, we think that there's probably other genes that modify that. And some of the strongest signals for that are genes that do DNA repair. So it may be that somebody with a repeat of 50, who you might expect would get sick earlier, at age 20 or 30, if they also have a genotype or a type of gene that is especially good at DNA repair, they may end up getting a later onset. So, we're investigating the modifiers. They're not always genetic. So for instance, environment plays a big part too, right? So in general, the things that are not good for your brain or your health, like substances or high blood pressure or, an unhealthy lifestyle that can affect your brain, that may bring on the disease, more quickly than just the CAG repeat would predict. So there's certainly other factors that modulate age of onset. But in general, that's on top of the larger framework that in general, repeat is going to dictate in general age of onset.
Yes, and that's a really important concept that the whole field and HDSA as well is investing heavily in finding out more about. We are getting a bunch of questions about whether this presentation will be available for viewing or emailing later. Yes, we are recording this right now, and we'll be posting it to YouTube and to our HDSA website. So we have some more questions about the cerebellum the cerebellum is related to balance, and HD affects balance. So some folks are interested to hear about, the relationship between the striatum and the cerebellum. If the cerebellum's overcompensating, then why is balance an issue? And does this cerebellum overcompensation really change how the brain is developing and making the cerebellum stronger?
Yeah, so it's definitely true that the cerebellum in general is involved in balance, but it's not just motor. It's also involved in thinking and behavior, and it grows together with the striatum. But I think what happens is that because the striatum is not grown appropriately, this cerebellum can essentially take over, and there's no motor symptoms at all until the disease onset. And we used to think about the disease onset coming when the striatum gets to a certain point that it can no longer function well, right? So we know this from the large studies like TRACK and PREDICT, that the striatum starts to decrease in volume 20 years before disease onset. That whole time, the cerebellum is compensating because, remember, disease onset is defined by motor symptoms. So instead of thinking that the disease starts when the striatum gets too small, we think that the disease starts when the cerebellum can no longer compensate. So what happens is that the cerebellum is trying to make up for the striatum, and finally it gets tired. And when the cerebellum gets tired and it can no longer compensate for the striatum, that's when you start to see the motor symptoms. And it's probably not just balance, right? So there's balance problems, but there's all sorts of problems that include cognitive and behavioral. So, both the cerebellum and the striatum connect to the frontal lobe, which is the areas of the, thinking and behavior that can be disrupted too. So, the cerebellum is keeping things together until essentially once it becomes too tired to help out anymore, that's when you get these symptoms, and you get the motor symptoms, you get the cognitive symptoms. And so it's really thinking about onset as when the cerebellum gives up rather than when the striatum gets too sick.
We have another question about possibly, if it's possible to go back to one of your slides. There's a graph showing the comparison looking at kids when they're 6 years old. And someone asks, is there a reason why the striatal volume is bigger in kids with repeats over 40 compared to those who don't have expanded repeats? So that looks like that red line group is already starting with a larger striatum there.
Yeah. It surprised us a little bit. But that's actually and I'm glad somebody asked about it, because that's like the most important point is right there, is that the earliest we could look at, 6 years of age, the striatum is already abnormal. And why is it too big? That's a good question. I mean, right now we don't know. Is it that there's too many cells? Is it that there's too many other stuff? Remember, there's not just neurons in gray matter, it's other cells too. So we don't know why it's too large. All we know is that it gets that way very early. So meaning that if we were able to push this back and look at 4-year-olds, look at 2-year-olds, it's likely that we would see that enlargement then.
So related to that, is there a benefit possibly earlier in life for someone who has more CAG repeats?
Yes, great question. So we see that exactly. So for people who have repeats, so if we look at people who are not going to get Huntington's disease, so they're not going to get it, so normal population, the more repeats in the Huntington gene, the better the brain development. Okay, the higher the IQ, the better the brain development. And that even within Huntington's disease, so if we look at the verbal fluency graph, So we see here that within Huntington's, those with the CAG repeats of 38 and 40 are definitely having better brain function than those with higher repeats. And in general, when we compare the 38 and 40 to people who don't have Huntington's, so they have repeats lower than that, this is the highest repeat. So there's certainly this notion that more repeats are beneficial to the brain, and that if you have relatively low repeats, but you're still going to get Huntington's, that is still beneficial, in particular in youth, because a CAG repeat of 38 may be, very beneficial to brain development, but development of Huntington's is likely to be much later in life, right? 70 or 80. And so
wow, so there may be some kind of evolutionary reason for having extra CAG repeats.
Exactly. And the evolutionary biologists have been telling this to scientists for a long time, that they believe these genes, and not just Huntington, but genes like Huntington, that the number of repeats allows for, better brain, and they may have been positively selected for human brain evolution. So the change from great apes to human is most dramatic in the context of brain growth. And so there's certainly this notion that the Huntington gene was potentially positively selected for human brain evolution, and that yes, that there is, up to some point, a significant advantage of CAG repeats. But then, of course, once you get to a certain point, then it becomes a disadvantage.
Someone else has asked what the Huntington gene actually does during development. How does it make development happen?
That's a great question. So, we've spent a lot of time understanding the mutant gene, and there's not a lot of science done on the normal gene. What we do know, it's a gene that helps transcription, meaning, essentially how genes are made into proteins, which is the very basics of how the cells develop. And so, how that gene affects when a gene is turned on, and it probably is genes that specifically affect growth and development of the striatum, a pretty an important region. So, to boil it down to kind of the molecules, this gene is involved in regulating developmental genes that have a direct effect on the volume and the trajectory of the striatum.
I want to respect your time, Peg. We still have about 3 or 4 more questions. Are you able to stay on for a couple more minutes?
Yeah,
or should...
I am. You bet.
Okay, wonderful. So the next question we have is if you've been tested for the gene, if you're over 18 but you are not yet symptomatic, can you still participate in Change HD if you
Absolutely.
know your status?
Absolutely. So if you are over 18 and you're at risk, you have a parent or a grandparent with Huntington's, you can participate. Anybody from 18 to, so if you've already been tested, that's fine. If you haven't been tested, that's fine. The only criteria is that you come from a Huntington's family, meaning that you have a parent or a grandparent with Huntington's disease. So whether you're tested or not, you can participate.
Great. We have a couple questions about late stage HD. Is it possible for someone within the age range who is highly symptomatic to participate? And we also have a question from someone who asks about their child, potentially about the child of someone who's symptomatic participating. So it sounds like if they're older than 6, they might be able to participate as well. But is it possible for a late stage HD patient to participate?
So the study is limited to those who are currently not symptomatic. And so for Change HD, you have to be, a diagnosis of Huntington's would exclude you from participating in Change HD. Now, if you were diagnosed with Huntington's before the age of 21, then we do have a protocol for juvenile onset that you could participate in. But if you are over the age of 18, I'm sorry, over the age of 21, and your diagnosis was over the age of 21, you would not be eligible. So
Okay.
let me just say that again. Change HD is for anybody who is at risk but not diagnosed with Huntington's disease.
Okay. We have a question about autophagy. Someone's wondering if you're looking at autophagy in this study.
So autophagy is something that would be evaluated at a molecular or cellular level, and our study is really systems level. So we're just doing, you know, clinical evaluations of symptoms. So we're not directly looking at that, but it's certainly something that could be and is evaluated by many other groups, looking at whether or not, you know, that process is something that's affected by mutant Huntington's, and whether or not it's affected by development. So. Okay,
great. Well, I guess the final question we have here is whether there is an active link for changehd.org. I think someone has already gone to tried to go to the website, and it looks like it's maybe not active yet.
We're working on that like today.
Okay. All right. So check back in a few days, everyone.
So the email is up, and the and definitely the phone is up. And I think what's happening is it's our previous website is Kids HD, and we're trying to change it to the Change HD. But yeah, someone beat us to the punch.
Okay.
They can get in touch with us the other ways. That'd be great.
Great. And finally, someone's wondering if you have published any papers on your previous work that are available to read.
Yeah, so in particular, the one with the striatal enlargement, so that one just came out a few months ago in neurology. So the Journal of Neurology has that, and the first author on that is Vanderplas, V-A-N-D-E-R, Vanderplas, P-L-A-S, and so yeah, so that's the one that shows the trajectories of striatal development.
And if folks want to write to webinar@hdsa.org, I can help you find that paper if you are looking to read it. So we've had a really great Q&A. Thank you so much, Dr. Nopoulos, for leaving all that time and answering everyone's excellent questions. And thank you again for this really great presentation. It was really clarifying for I think all of us. So thank you to everyone who attended today. Again, we will be posting a link to this recorded webinar later on. And I just want to thank everyone for attending, and thank you again, Dr. Nopoulos, for everything.
Thanks, Leora. Take care.
You too. Have a good day, everyone.